Good morning, students. Today we'll start photosynthesis second part. In the last class, we have discussed about the light reaction. In the light reaction, we have discussed about the cyclic and non-cyclic photophosphorylation. Now see the osmotic chemiosmotic hypothesis of electron transport system in Granum. The chemiosmotic hypothesis has been put forward to explain the mechanism of ATP synthesis, which is linked to develop of a proton gradient across membrane of the thylakoid. Thylakoid is a unit of Granum. Here the proton accumulation takes place towards the inside of the membrane, that is in the lumen. The proton that are produced by the splitting of water are accumulated inside the membrane of thylakoid. As the electron moves through the photosystems, protons are transported across the membrane. NADP reductase enzyme is located on the stroma side of the membrane. Along with the electrons from the acceptor, it removes proton from the stroma during reduction of NADPH plus proton. This creates proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. ATPase has a channel that allow diffusion of proton back to stroma across the membrane. This releases energy to activate ATPase enzyme that catalyzes the formation of ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient and ATPase. Energy is used to pump protons across a membrane to create gradient or a higher concentration of proton within the thylakoid lumen. See the ATP synthesis through the chemiosmosis. Cytochromes, movement of proton, and F0 F1 complex is there in the membrane of the thylakoid. As the proton moves, the energy is released and that results formation of ATP in presence of ATP synthase enzyme. How ATP and NADP is used? ATP and NADPH is the end product of light reaction. ATP, NADPH and O2 are the products of light reaction. O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the processes leading to the synthesis of food, that is sugars, carbohydrate. That phase is known as biosynthetic phase of photosynthesis or dark reaction. This process does not directly depend on the presence of light, but it depends on the products of the light reaction, that is ATP and NADPH, besides carbon dioxide and water. Melvin Kelvin after World War II worked with radioactive C14 and studied algal photosynthesis, which led to the discovery that the first carbon dioxide fixation product was a 3-carbon organic acid, that is phosphoglycic acid, that is PGA. He explained complete biosynthetic pathway and named that as Kelvin cycle or dark reaction. Further work of scientists led to the discovery of another set of plants in which the first stable product of carbon dioxide fixation was 4-carbon acid, that is oxaloacetic acid, that is OAA. Accordingly, those plants in which the first product of carbon dioxide fixation is C3 acid, that is PGA, is the C3 pathway, and those in which the first product was C4 acid, that is oxaloacetic acid, is C4 pathway. And on that basis, there are plants are grouped C3 plants and C4 plants. Photosynthesis, the process of photosynthesis starts when carbon dioxide is accepted by primary acceptor. The primary acceptor molecule is 5-carbon ketose sugar, that is ribulose biphosphate, RUBP. This is the second phase of the photosynthesis, dark reaction, where the fixation of carbon dioxide takes place with the help of RUBP. The next step in Calvin cycle, which have 3 stages, first is carbonylation, second reduction, and third is regeneration of RUBP. The Calvin cycle, Melvin Calvin and his co-workers proposed Calvin cycle. They showed that the pathway operated in a cyclic manner. The RUBP was regenerated. The Calvin pathway occurs in all the photosynthetic plants. It does not matter whether they have C3 or C4 pathway. Carbonylation. Carbonylation is a fixation of carbon dioxide into a stable organic intermediate where carbon dioxide is utilized for the carbonylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. The enzyme also has an oxygenation activity, so it is also called RUBP carboxylase oxygenase or Rubisco enzyme. And this enzyme is a protein, and that is present on the earth in a excess amount. This is the only protein which is present in the highest amount on the earth, that is Rubisco, RUBP carboxylase oxygenase enzyme. Second step of the dark reaction, that is the reduction. Series of reactions that lead to the formation of glucose involves utilization of 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for the reduction of per carbon dioxide molecule, which is fixed. The fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required for the removal of 1 molecule of glucose from the pathway. Third step of Calvin cycle is the regeneration of RUBP. Regeneration of the carbon dioxide acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. For every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. See the Calvin cycle. Ribulose 1,5 biphosphate is present in the plant. Carbon dioxide is fixed, combines with the RUBP. That step is carbonylation, and first stable product, that is a 3-carbon compound, phosphoglycerate, is formed. Then it goes for the reduction, where addition of hydrogen takes place and that PGA reduced to aldehyde, and then sucrose is formed. Then aldehyde again regenerated into RUBP in presence of ATP, and that step is regeneration. Now see the C4 pathway. Plants that are adapted to dry tropical region have the C4 pathway. C4 oxaloacetic acid is the first carbon dioxide fixation product. The plant with C4 pathway does follow Calvin cycle as the main biosynthetic pathway. C4 plants differ from C3 plants by having a special type of leaf anatomy. They tolerate higher temperature. They show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Anatomy of C4 plants differ from C3 by having large cells around the vascular bundles, which are called bundle sheath cells, and the leaves which have such anatomy are said to have crange anatomy. The bundle sheath cells may form several layers around the vascular bundles and have large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intercellular spaces. The pathway followed by C4 plants are called the Hatch and Slack pathway, as again a cyclic process. Because this cycle was first of all described by two scientists, Hatch and Slack. The tissues involved in the process of C4 pathway are mesophils and the bundles sheath cells. There are two types of cells, mesophil cells and bundle sheath cells of leaf. The process starts in mesophil where carbon dioxide is accepted by primary carbon dioxide acceptor, which is 3 carbon molecule, that is phosphoenol pyruvate PEP, and forms the 4 carbon compound, and that reaction is catalyzed by PEP carboxylase, that is PP case. Mesophil cells lack Rubisco enzyme. The C4 acids oxaloacetic acid is formed in the mesophil cells, which then forms other 4 carbon compounds like malic acid or aspartic acid in the mesophil cells. Malic acids formed are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release carbon dioxide and a 3 carbon molecule. The 3 carbon molecule is transported back to the mesophils where it converted to PEP, phosphoenol pyruvate again, thus completing the cycle. The carbon dioxide released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway which is common in all plants. The bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase oxidase, that is Rubisco, but lack PEP case. Thus, the basic pathway that results in the formation of the sugars, the Calvin pathway, is common in C3 and C4 plants. See the diagrammatic representation of the C4 cycle or Hatch and Slack pathway. Upper one is the mesophil cells, lower is the bundle sheath cells. The two cells are interconnected by the plasmodesmata, that is the small pores. Atmospheric carbon dioxide is first of all fixed with the PEP, phosphoenol pyruvate, and C4 acid is formed, malic acid is formed. That malic acid is transported to the bundle sheath cells where it breaks and forms C3 acids again and releases carbon dioxide. That carbon dioxide is now utilized for the C3 cycle, and that C3 acid is transported back to the mesophil cells where it again regenerates PEP to continue the cyclic process. Factors affecting photosynthesis. The external factors which affects photosynthesis includes ability of sunlight, temperature, carbon dioxide concentration and water. Blackman's law of limiting factor in the year 1905. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process of its quantity is changed. First factor, that is the light. At low intensity of light, there is a linear relationship between the incident light and the carbon fixation. As at higher intensity of light, the rate does not show further increase because other factors become limiting factors. Increase in incident light beyond a point results in the breakdown of chlorophyll and a decrease in photosynthesis. Carbon dioxide concentration also affects the rate of photosynthesis. Carbon dioxide is a major limiting factor of photosynthesis. It is important to remember that carbon dioxide is in very low concentration in the atmosphere, that is 0.03 to 0.04%. Increase in concentration up to 0.05% can increase carbon fixation, and increase beyond this level can be damaging over longer periods. Third is temperature. The dark reactions are controlled by the temperature because they are enzymatic. Light reactions are also sensitive to the temperature, but to a much lesser extent. The C4 plants have a much higher temperature optimum than C3 plants. The temperature optimum for a particular plant also depends on its natural habitat. Tropical plants have a higher temperature optimum than a temperate plants. Fourth is the water. The effect of water is more on the plants rather than directly on photosynthesis. Water stress results in closing of stomata and thus in reduced availability of carbon dioxide. Water stress also leads to wilting of leaves. Wilting of leaves means reduced surface area and reduced metabolic activity in leaves.